Welcome to Chem Help ASAP. This video describes a somewhat common problem that I recently encountered during a lab experiment. The problem is that some products are hard to tell from the starting materials, so it's not immediately obvious whether the reaction gave a product or just returned the original starting material. This mystery can take a little bit of effort to unravel. Here is a reaction that I recently performed. I also posted a video on it. It's a very simple conversion of benzyl chloride to benzyl azide with sodium azide. While the reaction is fantastic, you might see the problem in characterizing the product. Organic chemists rely heavily on NMR to characterize reaction products, and both the starting material and product have essentially the same NMR spectrum. 5 aromatic hydrogens and a 2-hydrogen singlet. This kind of problem is actually not uncommon. Reaction products often resemble the starting materials. After all, the reaction's products are made from the starting materials. Look at these two structures one more time. First, benzyl chloride. Second, benzyl azide. I'll refer back to each of them regularly in this video. To the left is the spectrum of the reaction product. The key signal is the CH2 singlet at 4.45 ppm. Is this consistent with the product? Absolutely. Benzyl azide should have a CH2 singlet much like this signal. Is this spectrum consistent with the recovered starting material? Again, absolutely. Benzyl chloride should also have a CH2 singlet just like this one. Now, there's no reason to think that this reaction did not work. But as a scientist, I prefer to have some more evidence for the reaction's success than it's just got to be the right product. Some NMR spectra simulations are available for a quick check of expectations. These simulations are often quite good, although not always. Here are predictions from a website called nmrdb.org. The CH2 group of benzyl chloride is predicted to be at 4.59 ppm, while benzyl azide is 5.05 ppm. My product, with a shift of 4.45, would seem to be closer to benzyl chloride than benzyl azide. That's not what I want to hear. ChemDraw, a chemistry drawing program, has a built-in NMR predictor in some versions. My version of ChemDraw predicts a shift of 4.64 for the chloride and 2.67 for the azide. There's obviously some disagreement in these predictions. Simulated spectra are often very helpful, but these two molecules, especially the less common azide functional group, are a challenge for some predictions. Before we keep talking about NMR, let's think about other techniques that we often use in lab to characterize compounds. With TLC, if two compounds have a similar RF value, you can often compare them on the same TLC plate as a 3-lane TLC plate. While it's possible for both compounds to have the exact same RF, this is rare. Even compounds with small differences in RF can often be distinguished as distinct on a 3-lane TLC plate. You can do the same thing with compounds that have similar melting points. Let's say the starting material and product of our reaction are both white solids with melting points of around 100 degrees Celsius. Many organic molecules fit this description or have nearly identical melting points. If you have a white solid with a melting point of 100 degrees, you might question whether you got the product or recovered the starting material. One possible solution is to perform a mixed melting point experiment. You can take a sample of the authentic pure starting material, mix it with your product, grind them together into a fine powder, and take a melting point. If the melting point decreases, then the suspected product and starting material are not the same structure. The melting point drops because the product is acting as an impurity in the starting material and depressing the melting point. If the suspected product were recovered starting material, then the melting point would be unchanged. In both the TLC and melting point examples, the identity of a compound is confirmed through direct experimental comparison to the properties of a known compound, the known starting material. We can use this same idea with NMR to solve our mystery with benzyl azide. Back to NMR spectra. On the left is a zoomed-in spectrum of the CH2 singlet of the product, which I believe is the azide, but it would be nice to be more certain. The CH2 singlet is very close to 4.45 ppm. To the right is the spectrum of benzyl chloride, the starting material. The CH2 shift for benzyl chloride is approximately 4.77 ppm, a bit further downfield. I'll note that both of these spectra were obtained with the same NMR solvent, deuterated DMSO. To confidently compare NMR shifts, all the samples should be analyzed with the same solvent. Well, what I obtained as a product does seem to have a different chemical shift and certainly seems very unlikely to be benzyl chloride. If it's not benzyl chloride, it's almost definitely benzyl azide. Still, it would be nice to have even more evidence that these are different peaks. The values of 4.45 and 4.77 are not very different. Chemical shifts in NMR can drift, although a drift of 0.3 ppm would be very surprising. Regardless, how can we be more certain? Just like with TLC and the mixed melting point experiment, we can mix both the product and starting material together in the same NMR tube and run a combined NMR spectrum. This new spectrum in the middle shows the azide sample spiked with some of the chloride. Now we can see that the two peaks are definitely different. To be clear, this does not prove that the product is benzyl azide, but it is clear that the product is not the starter material, benzyl chloride. There are other methods that we could have used to analyze the product. We could have compared the starter material and product by gas chromatography and looked at their retention times. We could have analyzed them by mass spectrometry. NMR, however, is normally the go-to method for answering these kinds of questions. While NMR is a fantastic tool for characterizing molecules, a single NMR spectrum is not always adequate for identifying a molecule, even a simple compound like benzyl azide. Sometimes you need to do a bit more work to be confident in the structural assignment. That's the end of this mystery. The reaction seems to have worked as expected, but confirmation required direct comparison of the product to the starter material. Please subscribe, like, or leave a comment. Take care.